Dr. Silva mentioned in his presentation tongue tip samples, and I wasn't familiar with that. Can you elaborate on that, please?
1, 2, 3. Can you hear me? All right, so tongue tips is sitting by Isadora Machado here, who is studying that as we speak. It's basically a procedure that's been used a lot, at least in Spain as we speak, and it consists of getting just the 1 inch from the tongue tips from the dead animals. If you think about the stillbirths or the suckling pigs that die in that first days of life, there's quite a bunch of those in any farm. And so you use that and you collect those in Ziploc bags all the way to 100, whatever fits in the bag. And then the exudate, the fluid that comes out of that, it can be submitted to the lab just like you do with processing fluids, and then test that we're testing for PRRS by PCR. And so that's another sample type that it's being evaluated as we speak. Probably have more soon to share.
And those, Daniel, do they collect those and then freeze them and send them, or how do they handle that? Can it be like a long-term storage, aggregate type storage sample?
It can be frozen, doesn't have to be. But if you freeze and thaw, that will help to generate a little bit more liquid. Right, Isadora, and that can be used just like your processing fluids. You may or may not freeze depending on how many samples, and depending on if you collected, Monday all the way to, you may want to collect as many as you want, submit at the end of the week. The more sample size, the better.
Great, good question. Other questions? Oh, hold on. Make sure we get it on tape here.
Giovanni, question for you. I think you mentioned that you thought that sow increase in breeding herd PRRS outbreaks trailed grow finish by 2 to 4 weeks, increase in that. Could it be the opposite, that grow to finish trails sow increases by the opposite time frame?
That's a good question. Thank you for that. If I did not explain correctly, that's is exactly that. You first start to see activity that's going on first in finishing sites, and then that follow-ups to happening in sow farms. So if you start to see activity occurring today, in 3 or 4 weeks down the road, you start to see activity in sow farms. So first happens in finishing sites and then in sow farms. It's the opposite of a biological way of sending piglets out, but seems like as you start to see that activity growing on in finishing sites, some level of pressure start to change in those regions, or some moving of people around and breach that finds out to move the virus across the system helps to do that.
Just one comment on that. One of the reasons may be because in the grow finish, as we know, it doesn't take much to do the chores in grow finish sites, maybe a couple hours, depending on the site. And what does that person do the rest of their day? They may come back and watch Netflix, or they maybe jump to the next site, right? And then the next one. It's not uncommon to find people taking care of 6, 7, 8 sites in a day. And so when they're moving site by site, there's a potential of bringing pathogens with that. And there will be a lot of other epidemiological connections between shared equipment, people, and so on and so forth, between the grow finish sites and the breeding herd. And that may be where we think that it's the virus gets to grow finish, it potentiates because of those epiconnections and shared labors, shared resources, and then it finds the way back to the sow farms. So one opportunity to kind of raise the bar in the industry on biocontainment there, on biosecurity, not only in the breeding herd, but also grow finish.
Other questions? Right down here, please.
Thanks. J.Q., you mentioned PRRS in brain. I didn't know if you're noticing like septic meningitis sites or signs with that, if those overlap. The reason I ask, the presence of strep or suspicion of strep often changes our antibiotic choice at that time. So I didn't know if you're seeing similar signs with like strep or not.
Yeah, so maybe clinically, I don't know. It's I always refer to Dr. Paul, So, yeah, so in our experimental study, we did not, include those co-infections. So maybe Marcelo can comment in the field situation whether you observed those, co-infection, whether other pathogens contribute to those.
Yeah, we in the research study there wasn't any signs of meningitis, bacterial meningitis or anything, so it was purely viral encephalitis. Now we have recently seen an increase in cases of PRRS encephalitis in the lab, and sometimes those are mixed infections with strep or GPS, but most often PRRS alone when it's viral, when we have lesions of viral encephalitis. And then the clinical signs, they're mostly ataxia and some lethargy. And if there are mixed infections, then that's when you started to see some classic paddling, lateral recumbency, that type of deal.
Well, while we're on this subject, Chris, I'm going to, Jake, ask you a follow-up question on that. So your results were certainly consistent with what we see in the field, or what in terms of how pathogenic, how virulent the virus is, and then what I was really interested in was how quickly, how easily it was transmitted. So in your experience over the many years, would the amount of virus that those pigs were shedding, would that explain why it shed so quickly, or do you think there's something else there, changing receptors, something else?
That's a tough one. We do not have a clear answer to that. So, but definitely the virus shedding levels, I think it, is one factor contributing to the transmissibility, because we can see this L1C variant, they shed more virus. Then at least, if you look at the dynamic changes, it's shedding high level than compared to others, it's earlier than others, So regarding the receptor, it's also complicated, because we all know for PRRS, we know the CD163, that is a major receptor, but look like some others could also serve as either co-receptor or whatever roles, So we have not really looked at that yet, but I can tell you that we also did the in vitro comparison of these 5 virus strains in vitro in ZMAC cells, in the same cell lines. We also observed that I did not have time to present that. We also observed that L1C variant replicate high titer than other strains, even in vitro. So now we are in the process of measuring the cytokines from those cell culture supernatant and also from the serum. We want to see if those virus strains, example L1C variant, will induce the high levels of cytokines, because the people, if you recently there's one paper, they have some criteria how to define that this virus strain is highly virulent. There's several 5 factors, clinical priorities, one, and also this cytokine level, they also considered part of that. So we are still in the process. Hopefully by the end of June, we will have those cytokine data. So to answer your question, it's lots of factors. It's just hard to define which one contribute more to transmissibility.
One quick comment on that, on the SDRS. We track the CT values, right, over time for the different pathogens. And Giovanni, you could correct me if I'm wrong, but this particular virus had the historically lowest CT ever observed, the database since 2007, maybe?
Yeah, that's correct. And that you need to remember a little bit of microepidemiological aspects of this. It started in Minnesota and Iowa, and those was the epicenter. If you look for the CT values for PRRS virus in those 2 states, when it was happening those waves, they dropped for the lowest levels that we ever had in the history that was detected. And as was I in Minnesota, we could not compare as the first wave of the 174 back in 2014 that was in North Carolina. But at this time, the city values was very low on the teams. So to change all the microepidemiological aspects of detection of fly state as Minnesota and Iowa, that for certainly there was something else going on there. And if you think as a city as a level of measure of pressure of infection, that could be a reason why you have more viral load in the environment.
Yeah, JQ, I wondered about that too. If you guys remember the picture, my favorite picture is the one where you got a picture of all 6 pens at 6 days post-infection. And if you look, you have the negative controls, and then you have the 3 older viruses. All those pigs are up, walking around. They all look normal. You have the 174, and you have the variant 1C, and those pigs all look like they're piled, they're laying there, they're thin, they've lost weight, obviously. And then you look at the microscopic, you look like they've got, 85 to 95% in the variant 1C and the 174 of lung damage. And then you look at the amount of virus in the serum. They're both, variant 1C is 1 log higher than 174, which is 1 log higher than those contemporary viruses. It just looks like with each new predominant virus that comes, there's just that much more virus, and we've just exceeded the pig's ability to be able to fight that off, right? And it's just amazing, and it really reflects, I think, in what we see with the pigs, and then unfortunately, what the practitioners and the producers are seeing with the mortality, right? And it's like, that was a little bit of, it looks like the long shot with the ivermectin, but that's the plead, with the pharma companies is like, hey, it'd be great to give us something, a 100% vaccine, but I'd be happy with something that would knock the log of PRRS virus back 2 logs, right, in the blood. That would get us back to viruses, 10 years ago. We know how to manage those. It's just amazing just to see how much more aggressive these more recent strains have gotten. So I was just curious to get your impact, if that was your takeaway from your study as well.
Yes, you summarized very well, But I do have one more comment about, you're all curious, people talk about this PRRS genetic classification system, what the term we should use, and ideally they wanted to have some term to refer to this PRRS, lineage, IFP pattern, whatever, that can reflect virulence, but unfortunately, we are not there yet, So here you can see that even for this lineage 1C, pretty genetically diverse there. I select the 2 subclasses within L1C. One I called L1C variant. Actually, then I said if we go with the sub-lineage, I called the L1C.5. Another is the non-variant, so I called L1C.1. You can see those, if you just refer to lineage 1C, you may show that all of the viruses within this lineage 1C will have the similar phenotype, virulence or whatever, but they are not. They are quite different. So back to this genetic classification, those are better. Now we still use OFF5. Whole genome sequencing, different story, and some group have proposed some classification system based on whole genome. But even so, that still cannot be correlated with this virulence, those phenotype. I just want to point it out, So, yes, science advances, but we still do not have those knowledge, because this PRRS is complicated. If previously people used the reverse genetic study that demonstrated that, these virulence genes could be across this whole genome, some in non-structured proteins, some in structured proteins. So it's just hard for that part. I wanted to point it out because even you get you got the 1C, you got L1C, but they are not necessarily identical. Can I make
Just to complement what JQ just said, from our perspective, when we talk to all these producers that had to go through this horrible experience, we can easily tell that even if you have the same 144L1C, the herds behave completely different, as you very well know. And one thing that we learned with Dr. Kikuchi when she did the case control study that got published is that there's something that we're not taking into account, which is co-infections, whether they are bacterial, whether they are viral. That's one, and maybe parity structure might be another one, right? So whenever we get the question of, is this 144 the same one as we're seeing in the Midwest, we'll say, well, from an RFLP pattern perspective, yes. From a lineage perspective, yes, but it may be a little bit different, right? So please do not think that the virus that you see on the other side of the country is the same one that your herd has until you get maybe a whole genome sequence compared, right? Because it's just very, very dangerous to say, I've got the same virus that it's circulating in the Midwest, right? So I think we'll just keep saying it over and over and over just because people tend to generalize that this virus is really bad, when in reality, it may not be that case in all of the herds. So just a word of caution.
Yeah, very good. Question over here.
Sarah Meixel with the Pig Site. I had a question, and I know Dr. Silva is not here with us, but I was hoping maybe somebody on the panel could answer. What he was showing, I guess, with how you can use your existing data, right, to predict, not predict, but to monitor real time, I guess. Is that being done in the field right now? Is that, are people using that approach, I guess, to monitor their herds with success or without success? I guess I'm just curious. It looked really easy is probably not the word to say, but when you're looking at it backwards, which is what I'm assuming they were doing, it was pretty easy to see those trigger points, right? Where you're getting to a point where you're going to see it, or something's happening, right? That's out of the norm. And I was just curious if that's happening real time with production herds right now.
Yeah, good question. We are excited about that. We see the production systems are moving the last, I think, 5 years, and now more intensively now as we speak, moving to their production data from their legacy software to somewhere in the cloud. And regardless of the platform, if it's in the cloud, and if it allows some API with our servers, we can. It is possible, and some producers are doing that as we speak, to monitor those numbers as they come. Right? You don't have to monitor all the production data, like Dr. Silva said. Just a few key performance indicators. You think about aborts. If it's an ESF system, some metric of feed disappearance. Right? You may want to monitor neonatal losses. So if you pick 3, 4, you're good to go because they are all auto-correlated between themselves. So the short answer is yes, especially those production systems that already have their data on the cloud. And if there is any way to get access to that real time, and again, as we move forward, it's getting easier and easier to do that. It's a way to monitor your herd on a daily basis, right?
Is that kind of information, Daniel, going into like some of the things that Giovanni's studying? I guess is individual data like that part of what is captured by some of the reports that you guys do?
So I'll just repeat because it was not on the microphone, right? If I understood right, the question was, is this data connected with other data streams?
Right? Is that data being, any of that kind of cloud data, are producers sharing to that level? Or are they just sharing kind of, yes, we've had an outbreak and this is the information that we have. So I've been receiving what Giovanni puts out, and it's a lot of information, right? It's a lot of data. And I guess I was just curious how that data comes in. Do you guys get kind of a little bit more nitty-gritty with that? Or is it more information that's coming in that's like, just we've had an outbreak, this is the type of outbreak, or this is what we're seeing? So I'll stop talking now. You guys can go.
When you say the data that Giovanni is collecting, it's kind of challenging because he collects a bunch of different types of projects. If you're referring to the SDRS, that his first presentation, that's not connected, because that's all aggregated data without any premises information stripped from the data, right? So that's all in the state level, what's going on throughout the
that's
U.S.
just all coming in at a different level than like a producer type level
Right.
data.
Now, if for the second project where he collects whole genome data, that's really on-demand type of deal, type of thing, right? Producer goes through an outbreak, wants to further understand, could hook up with Giovanni to get that whole genome analysis done, and then partner up with the production data, some other stuff, to be able to kind of get herd-level metrics associated with the whole
Got
genome
it.
data.
Okay, sorry,
that
I'm getting
help?
confused. Yeah, it does.
All right.
I just wasn't sure what kind of data was, I guess, being fed into some of his reports.
What was your last question?
I just was asking what kind of data was really being populates the reports that you put out, right? That I think it's weekly. Is that right? Weekly.
Yeah, so that's a pretty good question for the swine disease reporting system. So we have this network of laboratories that is Iowa State, University of Minnesota, South Dakota, Kansas, and Ohio. So they share with us on a daily basis anonymized data for testing and testing results from these PRRS, influenza, PD, and Mycoplasma pneumoniae, PCV2 also, on anonymized way that is producer, client, those farm, it's everything anonymized. We receive from these 5 labs and we anonymize, and then we put these reports out on a daily basis. So if you don't get the report, you can subscribe for the team to send that out. But online, there is dashboards that are daily updated with the new results that come in.
Okay. Very good. Thanks so much.
Other questions? We still got a few minutes yet before our break.
I got one quick for
Okay.
Dr. Holtkamp. There are one quick question. Sometimes when we talk with different people sometimes interpret the term stability differently. Can you just remind us what is stability, and is it different mycoplasma and PRRS, or is it the same meaning?
Yeah. Thanks for asking that, actually. So, yeah, when that term specifically means the ability to wean negative pigs on a consistent basis, right? So that works for PRRS, it works for PED virus, but when it came to mycoplasma pneumonia, that didn't make sense any longer, right? As a way to define a herd based on their myco status. And so what we ended up doing on the myco herd classification was to use the term controlled and uncontrolled, right? So rather than stable, purposely to try to retain that definition for its true meaning for PRRS. But you're right, I've kind of given up on, I used to correct people like a old English school marm, but I kind of given up on that. But that is right, that's what it means, so.
Oh, yep.
Just a general question. Is there more work being done on the environmental survivability of these 144L1Cs versus other strains? I think there's recently a report out of Minnesota kind of saying that maybe the 144L1C survives longer in the environment. So just curious if anyone has any other work going on in that area.
So besides that report that you may have seen from Dr. Goyle's lab and Dr. Angie Quiñones, there's some others that are very preliminary, but they're kind of pointing towards they're in the same direction, right? So that's one thing. The other thing is that previously, before all of this 144L1C, there was some other work done looking at different RFLPs. And I think this was 2016, 2017, and there were differences among RFLPs that hasn't been published, though. So we can see that there might be differences among families of viruses, if you want to call it that way. So that's one thing. The other thing is, recently, Dr. Marcelo and I, we went to some farms that are receiving 144L1C positive pigs, growing pigs. And we did some environmental sampling, and it was amazing to find some CT values of, I can't remember Marcelo, but he has the numbers, but there was a low 30s, high 29s on the surfaces of those farms. Door handles, right? Or if you see some of the floor just right before you leave the barn, you would find quite a good amount of viral particles there. So it's not only the concentration that we see within the pig, but the shedding, the survivability, and then outside the host, that it's bringing all this into focus in the sense that we may be moving it around ourselves without knowing it, or trusting that we're doing all the processes right. So I think it goes all into the same formula, but it's telling us I'm putting out a lot of virus, and on top of that, it's surviving well outside. So it's concerning.
I'll add to that, and Mafalda's over there warming herself in the window. But I would put her on the spot, and I may still do that, but Dr. Silva, Mafalda, and I, or several others, just did completed a study looking at different times and temperatures and look specifically at the lineage 1C variant. And Mafalda, I'm going to summarize, and you correct me if I'm wrong, but there wasn't a lot of difference, right? I mean, a little bit, but it was not, I think, what was it? The other virus was a 184?
Minnesota 184.
Yeah, Minnesota 184 compared to head-to-head to the lineage 1C variant. And so at least its ability to survive temperatures at different times. And the range we looked at was from 20 degrees Celsius up to, I can't do the conversion, 122 degrees Fahrenheit. Room temperature to 122 degrees Fahrenheit. And so not a lot of difference there. But is that right, Mafalda? Okay, she's nodding her head yes. So.
Can I say something?
For sure, absolutely.
So it's not related to today's presentation, but since there's some producers here, I'd like to mention some, things one of my graduate students recently did. I believe some of you may request the PRRS virus isolation and then forward isolate to a third party to make the autonomous vaccines. It's a pretty common practice. I just want to remind you about something. So, for the PRRS virus isolation, most of the labs, they just do the virus isolation, then based on the cytopathic effect or fluorescence staining to tell if it's positive or negative. If it's positive, they forward isolate to the party for autonomous vaccine production, but they do not sequencing isolate compared to the virus present in the clinical samples. So recently, one of my students compared this over 1,000 clinical samples and PRRS isolated in MAC145 and ZMAX cells to see. And I can tell you that over 97% of those clinical samples and 2 cell line isolate that match each other. However, roughly 2.4% of those MAC145 isolate have the different sequences when you compare it to the virus in the clinical samples. In contrast, the ZMAX isolate, they all match. So for those MAC145 isolate that do not match with the clinical samples, we further did the whole genome sequencing and find out that those samples in the clinical sample actually have the co-infections. So let's say if you have the predominant strain, the wild type strain, you have the minor strain. Example is, for this case, is the BI-MLV vaccines. So if you just sequence the clinical samples, you will find the wild type strain. However, when you do the isolation in MAC145 isolate, those minor vaccine viruses outgrown compared to wild type, you isolated the vaccine virus. My point is that if you do not sequence those, you forward those isolate for autonomous vaccine production, you actually just based on the vaccine virus, not your farm-specific wild type virus. So I mentioned here, I just said, especially if you use the MAC145 isolate for autonomous vaccine production, be sure to sequence it compared to your clinical samples before using it. That's the one point I want to share.
Very good point, JQ. Any other questions? Any other questions for the panelists? Any other questions from the panelists? All right, got one right down there, please.
Good job, all of you guys. Giovanni, you were talking about how when you detect more than two viruses in the same farm, how that impacts the productivity. Did you have the chance to investigate or to look into the data to see what were the characteristics of those farms that had more than 2 or 3 viruses circulating in those farms? Is that more just determined by location, or was there something else?
Hi, Marcelo. Thank you for the question. That's a good one. When we start to look into that, the characteristics of the farm seems like they did not matter in terms of number of strains that you got. And why I say that is because there was farms that, well, they decided to eliminate the virus from the herd, and they did a closure and went through that process, and we find more than 2 strains there. But there was else farms that were naive, status 4 of the ASV, that broke with PRRS and went through and farmed more than 3 strains than the farm. So that's kind of eye-opening, and you start thinking, well, maybe when we go through a PRRS virus outbreak, we're not introducing only one PRRS virus strain in the herd. We may be introducing more because we have more that are circulating. That could be the case. So back to your question, was not clear characteristics of farms to determine if you're going to have one or more strains. The thing that was kind of interesting was, well, if you have more PRRS virus strains circulating, you also have the opportunity to generate new strains by recombinations and mutations on those farms else.
And Giovanni, when I was wondering about that too, when you guys did those, how many samples were you making that determination? Was that on a single sample that you'd find more than one virus, or a series of samples? How did you make that determination on how many viruses you had in the farm?
So that's a good question that goes back a little bit on the materials and methods of this study that I did not explain. So what's herds that break with PRRS and they adopt measures to eliminate the virus from the herd, we look to get this reference strain at the time of the break, and then we use processing fluid samples that are sample type down the road. And that was around 4 weeks after the break, the first sampling type, around 10 weeks, and then we froze out the samples until the herd achieved low prevalence, and we went backwards and select the samples with CT lower than 30 and sequenced those ones. So it was an overtime following of those herds. And one thing that was interesting was when you go down the road to the elimination process and the predominant strain starts to be less predominant in the herd, then you start to find these additional strains that kind of popping up and opportunity to appear with some, maybe there are some groups in the farm that they are clustering and circulating, come to the farm house and you are able to find those.
Okay. And what does a recombinant strain look like? How can you tell on a whole genome sequence it's a recombinant strain?
It's variable. If you remember that image that I showed, well, if you stretch this virus, you have 15,000 base pairs aligned, and you have recombinations that they grow. Any of those points can occur. There is no hot spot today that you can say, oh, they are going to happen here or there. They are, I have seen all over the places. And they can take one strain, and that could be a wild-type strain with another wild-type strain. They can take one wild-type strain with a vaccine strain, or they can take multiple wild type with combinations of vaccine or whatever. So the point is, well, we have a lot of wild type strains out there, and maybe we are missing a lot of those opportunities to detect recombination because we don't have a bank with full set of wild type strains that we have today. On the other side, vaccine strains, we have a handful, and they are easy to find because we just blast against them and we found those. But we miss the wild type ones for sure.
Very good. Any other questions? All right. If not, what we will do is we'll go ahead and adjourn till 2:55. Looks like we've got some refreshments over there. We'll start promptly at 2:55 with our breeding herd panel. Thanks for your attention. Let's thank our speakers.